Welcome everybody. So today we're going to be looking at yet another DNA manipulation technique that we've again got from bacteria as well. They keep coming up. So we're going to look at how we can make recombinant plasmids using bacteria cells. We're going to look at a really specific example of insulin and I'll elaborate now. So what is DNA cloning? So this process can be referred to as a lot of things: DNA cloning, recombination, bacterial transformation. It has a range of different names, but they all mean the same thing. So we've looked at how to replicate DNA through PCR, how to visualize it through gel electrophoresis, which is fine. But what if we actually want to use that gene to create a protein? So we don't just want to visualize it, we actually want to use it and we want to create a protein product. This is when we have to do something called DNA cloning. So if we have a particular protein of interest, for instance, insulin is the example we're going to use today. So let's say that we want to produce a mass amount of insulin. We need to make many copies of that gene to produce many proteins. So originally we were getting our insulin from pigs to help treat people with diabetes. So diabetes is a condition in which you can't produce your own insulin. So we were getting it from pigs, and hopefully you recognize the ethical problems with that and the poor piggies. So we developed technology to make this process better. And we can now use bacteria to replicate this gene for us, and we can mass produce insulin protein to help treat diabetics, which is really cool. So why are we using bacteria yet again? So bacteria are really good for this process because they have these things called plasmids. So our DNA is tightly wound up into chromosomes in our nucleus, whereas bacteria, they don't have a nucleus. They have a circular DNA chromosome, and they also have these things called plasmids. So these plasmids are really easy to manipulate, and they're really quick at replicating. So that's why we use these, because we can make many genes and many proteins in a really short period of time. So we can manipulate these plasmids to synthesize our proteins for us. So what we can do is we can insert a gene of interest, and then the plasmid replicates, and then this insertion makes the plasmid called a recombinant plasmid. So this is where all the different names come from. So the term recombinant plasmid just means a plasmid that has been inserted with a gene of interest. So when the bacteria take up this recombinant plasmid, so we do this outside of the bacteria and then we pop it back into the bacteria, and this is called bacterial transformation. So once we have the plasmid and the gene has been inserted, we insert it back into the bacteria, and these bacteria are said to be transformed. So that's where the other name comes from. And of course, the most common example, like I said, is the mass production of insulin protein to treat diabetes. So that is how we are currently making insulin protein today. So let's go through the steps of how we actually do this. So the first thing we need to do is we need to make the recombinant plasmid. So bacteria have these plasmids, that's all very well and good, and they're really good cloning vectors because they self-replicate, they're small, the bacteria take them up really easily, and we can really easily insert things that we want in there. So once we've isolated a gene of interest, example insulin gene, we grab this plasmid vector, we grab a restriction endonuclease. Okay, so remember our endonucleases, they're our molecular scissors, and we grab a DNA ligase. So remember, they're like our DNA glue, and they're the three things that we need to create a recombinant plasmid. So the plasmid that we choose will have the restriction endonuclease sites. Okay, so obviously we need something to cut. So the plasmid that we choose is going to have the restriction enzyme recognition site, for instance here. Okay, so it's going to have that. We want sticky ends because remember from our last couple of videos, sticky ends are more beneficial for gene insertion. So we're going to find a plasmid with that. We're going to find, or we're going to create a plasmid that has antibiotic resistance gene, and I'll elaborate on why that's important a little bit later on. It's going to have an origin of replication, and it's going to be able to take up a reporter gene, which again I'm going to go through a little bit later on. So what happens is we use their restriction endonuclease to cut the plasmid, okay, and then we insert whatever we're wanting to insert, whether that be insulin gene or something else, we insert that into the sticky end part of the plasmid, and then we use ligases to rejoin the plasmid together again. So we've got our fully formed plasmid with our gene of interest, which is ready to be taken up by bacteria. So that's how we're using all the tools that we've learned about to make a recombinant plasmid. Then what we need to do is we need to transform the bacteria. So we've got this plasmid, we now need to have the bacteria successfully take it up so that they can start replicating it. So bacteria will usually naturally take it up if it's free floating, but we can use a couple of different methods to help us to make sure that it takes it up. So we can use something called heat shock. So when we heat the plasmids, it makes the bacteria more permeable and more ready to uptake the plasmid. Or we can do something called electroporation, which is a similar concept. We use an electrical current to make the plasma membrane more permeable for the plasmids, for bacteria to take them up. So we can speed this process up. Oftentimes we will also, when we're making a recombinant plasmid, we'll insert a reporter gene. So a recombinant reporter gene, so basically what a reporter gene is it's a way to make sure that the bacteria has transformed correctly. Okay, so a common reporter gene we can use is antibiotic resistance gene. So if we put antibiotic resistance gene in our recombinant plasmid and then we place our transformed bacteria onto a plate of antibiotics, we know that the colonies that survive will be successfully transformed. So how do we know? So if we've taken up the gene, we've also taken up the antibiotic resistance gene. And if we have that gene, we are resistant to antibiotic, which means that those colonies will survive. So if for whatever reason the bacteria has not taken up the gene or the plasmid, they will die on this colony because they do not have that antibiotic resistance gene. So that's one of the many ways that we can make sure that our transformed bacteria are transformed. And then we will take all the transformed colonies and then we'll mass produce the protein. So this is the example that I want to focus on and that Vika also wants you to focus on. It's looking at this recombination process, but using insulin as a specific example. So insulin is a hormone that regulates blood glucose levels, and people with diabetes do not produce it enough naturally. So therefore we need to artificially give them this insulin protein so they can regulate blood glucose levels. So this insulin protein has a quaternary structure. Okay, so remember from the very first video you watched what that means. So a quaternary structure means that it has multiple or one or more than one tertiary structures joined together. So insulin is actually composed of two separate tertiary structures. We've got the alpha structure and we've got the beta structure. So because it's a quaternary protein, that means that we have to go through two different stages to make the fully formed insulin. We need to make the alpha protein and we need to make the beta protein. And then at the end, we will transform the two. So that's why you'll see that they've gone into two different branches here. This one is the alpha unit and this one is the beta unit. So we have to do two sets of recombination for every time we want to produce this insulin. So what we do in this specific example is we will take two plasmid vectors, okay, because we need to do two different transformations, and we prepare them with two different reporter genes. So we use the antibiotic resistance reporter gene like we did in the other example, and we also use something called a LAC-Z reporter gene. So the antibiotic resistance is the same as the other example. So if it has successfully taken up the gene, that means it's also taken up antibiotic resistance gene and will be able to survive on the antibiotic plate. What LAC-Z is LAC-Z has the same restriction site as the gene of interest. So that means that if LAC-Z and the gene of interest are successfully cut and taken up, this LAC-Z reporter gene should be cut. So what that means is that LAC-Z is a natural, it produces a blue compound. So if LAC-Z is being cut, then there should be no blue compound. So that means that we're basically doing two different reporter genes to just double check that our plasmids have been transformed correctly. So what we're going to do is we're going to use an endonuclease with a specific recognition site, and we're going to cut that plasmid, and we are going to insert our gene of interest, plus our antibiotic resistance, plus our LAC-Z reporter gene, and we're going to use DNA ligase to put it all back together again. Okay, so we should have, we'll hopefully have a successfully put together plasmid. We're going to use either heat shock or electroporation to help speed up and ensure that our bacteria successfully take up these plasmids. And then we're going to use our reporter genes to make sure that it has been efficient. So we're going to place our bacterial cultures on an antibiotic plate. Okay, so our first stage of, making sure that everything is correct is we're going to put them on antibiotics. And if they have been successfully transformed, they will survive. The other way that we're going to figure out whether they're successful is if they're colorless or not. So if the gene of interest has been successfully cut and pasted into the plasmid, that means that the LAC-Z reporter gene should also have been cut. So because it's been cut, it should not work. So there should be no blue fluorescence. They should be colorless. So that means that if our colonies die or if our colonies fluoresce blue, they've not been successful and we can remove those colonies. So the ones that survive the antibiotic plate and the ones that are colourless have achieved successful transformation. And we do 2 steps just to make double sure. So once we've found out that they've been successfully transformed and the bacteria has taken up these plasmids, we can put them in the right conditions to ensure that they successfully transcribe and translate a mass production of insulin protein. So once we've done this for the alpha unit and the subunit, at the end, we will take all of our alpha units and all of our beta subunits, and we'll put them together in their quaternary structure, and then it will be a functional human insulin protein that we can use to help people with diabetes, which is lovely and much better than using poor little piggies. So in terms of DNA cloning, using insulin as the example is going to be most beneficial for your VCA exams. So just remembering, just a summary, we use endonucleases and ligases to cut and paste the gene of interest. We use antibiotic resistance and LAC-Z reporter genes to make sure that the bacteria have successfully been transformed. And then we can encourage transformation using heat shock or electroporation. And then we will culture them and see whether or not they've been successful using our reporter genes. And in the context of insulin, we'll do this twice to produce both of the subchains, and we will combine them at the end to make a functional protein. So hopefully this has helped. As always, bring your questions to class and I will see you there.